Okay, so this is going to be the second part of our meiosis mutations lecture series. So if you did not watch the meiosis lecture, please go back and do that before watching this one. So this is chromosomal mutations. So remember back in our mutations unit, we talked about mutations on a specific gene. And I told you then chromosomal mutations were coming. So that day is today. So we're going to talk about the second type of genetic mutations, chromosomal mutations, which lead to chromosomal disorders. And these are mutations that cause disorders that offspring, say, are born with. So what happens in chromosomal mutations, it sounds like just like what we're talking about. You're going to have a result, a change in the number or the structure of a chromosome. So what does that mean? It could change the locations of genes on a chromosome. So maybe a chromosome gene that's supposed to be towards the bottom of the chromosome is now in the middle or at the top, and that can cause problems. And it's going to result in the DNA sequence being changed, of course. And so just like in genetic mutations, if it codes for the same amino acid, there's not going to be a change. But often when you're talking about a number or pieces of a chromosome missing or in the wrong location, it does code for a different amino acid that it's supposed to. And this is going to result in a phenotypic change. So when we talk about a phenotypic change, what are we talking about? We're going to talk more about Punnett squares. You'll have a nice flashback to 7th grade. But phenotypes, those are the things that you see. And I'm putting that in little quotation marks with my hands because in some cases, phenotypes you can see. All right, so physical traits like hair color or hair type or eye color, those are phenotypic traits that you can see. But phenotypes also code for traits that you can't see with your eyes. So for example, your blood type is genetic. Now you can't see that with your eye. Even if I put it under a microscope, you wouldn't be able to tell me what blood type you have. You'd have to do a test for that. So that's a physiological trait that is tied to genetics. So chromosomal mutations can form or result in chromosomal disorders, and they are named for the chromosome that is affected. So again, here's your AKS. So we are going to be focused on two types, monosomy and trisomy. And so you are going to want to know what they are, how they happen, and what is the result there, okay? So when we talk about chromosomal disorders, when things go wrong, non-disjunction, what does that mean? It means an error in meiosis. And what happens is the homologous chromosomes, so remember that chromosome that comes from the biological mother and the chromosome that comes from the biological father, when they fail to separate correctly during meiosis. And so sometimes you end up with more chromosomes than you're supposed to have. Sometimes you end up with less chromosomes that you are supposed to have. And sometimes you end up with chromosomes that are longer or shorter, so only a portion of them have been affected. So we're going to watch a clip about non-disjunction, and they're also going to introduce you to some other things like karyotypes. So to figure out what type of non-disjunction has occurred, here's what you need to ask yourself. What would the resulting zygote, that fertilized egg, what would it look like? Would it have more than the correct number of chromosomes? Would it have less than the correct number of chromosomes? If a mutation hasn't occurred, then you'd have the normal number of chromosomes. And on your test, you will need to be able to discern what kind of non-disjunction has occurred, either through a description in words or through diagrams. And we're going to go through the diagrams after the video and take a closer look. So let's watch the video.
Hi, bio people. We're going to follow up on our discussion of mitosis and meiosis by talking about chromosomal abnormalities. These are all terms that you should be familiar with by the end of this screencast. And we're going to start off by talking about karyotypes, and then move on to non-disjunction, Down syndrome, and something called amniocentesis. Remember that DNA is coiled up very tightly into something called a chromosome. A segment of the DNA that codes for a specific group of proteins is called a gene. You have a lot of genes that all determine different traits that you have. So for instance, you have a certain number of genes that code for how tall you are, what color hair you have, or what color eyes you might have. Somatic or body cells are diploid. Diploid means that they have 23 pairs of chromosomes. This is different from sex cells like sperm and eggs because sex cells are haploid. They have 23 single chromosomes rather than having 23 pairs. This means that body cells have a total of 46 chromosomes, whereas sex cells have a total of 23. This is what is called a karyotype. It shows all of the different chromosomes that were inside one person's cell and matches them up with their pair. Because we have 23 pairs of chromosomes here, we can determine that this actually came from a body cell rather than from a sex cell. Each pair is made up of 2 sister chromatids. 23 pairs in all. Now notice this last pair, the 23rd pair, has 2 that look pretty different from one another, which is different than any of the other preceding pairs. This is because the last set of chromosomes, the XX chromosome or the X and the Y chromosome, determine the person's gender. If the person has an X chromosome and a Y chromosome, they are biologically male. If they have 2 X chromosomes, they are biologically female. Notice how the Y chromosome is very short and stubby compared to the X chromosome. If we look at this other person's karyotype, we can see that their X and Y chromosomes look a little bit different. Here we can see that they have 2 X chromosomes and no Y chromosomes. And so this tells us this person is female. It's very important that all of your body cells should have 23 pairs of chromosomes. If there's a different number, too many or too few, then you have what's called a chromosomal abnormality. For instance, if you have an extra copy of chromosome number 21 and have 3 copies instead of 2, we call it trisomy 21. This is what causes Down syndrome. Before giving birth, parents can choose to have their baby undergo something called an amniocentesis. This is where they take a small sample of the cells and fluids surrounding the baby and test it to see if it will have chromosomal abnormalities. Things like trisomy 21 will usually show up on an amniocentesis before the baby is born. However, this also raises a number of ethical questions. Should people then be allowed to terminate the pregnancy if they decide that they are not able to care for a Down syndrome child or a child with a serious chromosomal abnormality? This is an ethical question rather than a scientific question, but it's important to keep ethics in mind as we continue to discuss this topic. Remember that in mitosis, a diploid parent cell splits into 2 identical diploid daughter cells. In meiosis, we go through 2 rounds of division, transforming a diploid parent cell into 4 different daughter cells, each of which are haploid. Each of these has genetic variation thanks to 2 procedures called crossing over and homologous recombination. Every single daughter cell is different from the other daughter cells, as well as being different from the parent. Meiosis consists of 2 rounds of cell division, each one consisting of PMAT, prophase, metaphase, anaphase, and telophase, just like in mitosis. However, since there are 2 rounds of it, we go from having a parent cell to having 4 haploid daughter cells. So we go from having a parent cell that looks like this, with 2 red chromosomes and 2 blue chromosomes, to having 4 daughter cells, each of which have 1 red and 1 blue chromatid. This is what happens in normal cell division. However, we're now going to take a look at what happens when it goes abnormally. In a nutshell, abnormal chromosome division occurs when you wind up with the wrong number of chromosomes in one of the resulting daughter cells. Instead of having 1 of each color, you wind up with too few or too many. Before we dive into the abnormal procedure, please remember this is a simplified diagram. You have 23 pairs of chromosomes, but here I've only done red and blue to keep things simple. Normally, half of the chromosomes wind up on one side and half of them wind up on the other side. However, sometimes this doesn't happen, and we wind up with too many chromosomes on one side and too few on the other side. So what if the cell split like this instead? This means that after meiosis 1 and 1 round of cell division, we would have 1 blue chromosome on the left-hand side and 3 chromosomes on the right-hand side. Usually the problems with cell division occur during anaphase, when the chromosomes are not pulled to the correct side of the cell. If we were to proceed through meiosis 2, even if everything went perfectly from that moment forwards, we would wind up with all 4 daughter cells having an abnormal number of chromosomes. Instead of each one having 1 of each kind, you have some that have too few and some that have too many. So this is what happens if the problem occurs during meiosis 1. Now let's see what would happen if the problem occurred during meiosis 2 rather than meiosis 1. We're going to assume that the first round of division, meiosis 1, prophase, metaphase, anaphase, and telophase 1, went perfectly, and we wound up with exactly the right number of chromosomes in the first round of cells. If cell division proceeds correctly from here, we'll wind up with 4 haploid daughter cells, each with the same number of chromosomes. But if one of them divides incorrectly, we're going to wind up with an incorrect number of chromosomes on one of the daughter cells, or possibly even 2. So let's just say that this cell right here on the left is where the problem occurs. Usually the problem occurs during anaphase, where 1 chromosome failed to separate into 2 sister chromatids, and so too many of them are on one side and too few are on the other side. This is what the daughter cells would look like after both rounds of meiosis if there was non-disjunction. Non-disjunction simply means that the sister chromatids failed to separate correctly. Even if this cell over here on the right divides perfectly, you still have half of your cells that are not correctly formulated in terms of the number of chromosomes that they have. Non-disjunction is much more common in older individuals. So the older someone is, the more sticky their chromatids become and the less likely they are to separate. This is why older parents are more likely to have children with chromosomal abnormalities. Non-disjunction can occur either in meiosis 1, meiosis 2, or in both. However, we can see here that having it occur in meiosis 2 is slightly better than having it occur in meiosis 1. When we look at meiosis 1 non-disjunction, every single daughter cell down here has an incorrect number of chromosomes. However, if the first round goes correctly, only half of them have the wrong number of chromosomes, and so the resulting child would have at least a 50% chance of having no chromosomal problems whatsoever, but a 50% chance of having something like Down syndrome. I think that's all for now. I'll see you all in class. Make sure you bring questions.
Okay. that's a really important video. So it's going to be listed separately under my content, but you can always go back and take a look at that. I think she gives a really good explanation for what happens in the comparison between the normal gametes versus the abnormal gametes. So again, we're going to focus on what the zygote would look like, more, less than the correct number of chromosomes. That will help you decide what type of non-disjunction has happened. So you can always go back and take a look at these diagrams to help you. You're going to see diagrams like this on your test, and you will have to do a comparison and sort of figure out what has happened. And so in some cases, you got to pay attention to what you have starting within that germ cell to determine what the normal number of chromosomes should be in the haploid cells. So remember, when we're talking about meiosis, we are going to be talking about a germ cell that's going to form either 4 eggs or 4 sperm. So these are sex cells down here at the bottom, and they should have the half of the number of chromosomes that you see at the top. So let's take a look at monosomy. So mono means 1. Okay, and it refers to the fact that only 1 chromosome from the pair are in the fertilized zygote. So how does that happen? So let's take a closer look at that same diagram. So let's look at the normal. So the normal egg or the normal sperm should have 1 chromosome. And I'm going to point out that these are really simplified examples. For example, humans have 46 chromosomes, meaning in their sex cells they have 23 chromosomes. That would be a lot to squish into an egg or a sperm. And so these examples that we are using, you're going to see 1 or 2 chromosomes just to make it easier. But we're going to come back to that in a second. So in this particular example, the 4 eggs or the 4 sperm should have 1 chromosome inside of them. But if you look over here at the circled parts, you can see that there is non-disjunction that has occurred. And they're giving you 2 different examples. And we've ended up with all of the chromosomes here. And so when they split, they got allotted to these 2, let's call them these 2 eggs. Okay, so the other 2 eggs didn't end up with any chromosomes at all. So then when, let's say, egg letter A is fertilized by a normal sperm, okay, and they came together and formed the zygote, they wouldn't have the correct number of chromosomes, right? Because this donor egg doesn't have any chromosomes to give, all right? And that's going to cause some problems. Let's increase the number of chromosomes for a second. Let's pretend that a normal, let's take a human example. So a normal human egg would have 23 chromosomes. But let's say that chromosome number 7 in the egg is missing due to a chromosomal disorder or a chromosomal mutation. And so a normal sperm with 23 chromosomes fertilizes this egg that is missing chromosome number 7. And so all of the other pairs, so pairs 1 through 6 and pairs 8 through 23, are going to form their homologous chromosomes, 1 from the mom, 1 from the dad, normally. But chromosome number 7, remember, the egg didn't have any to donate. So chromosome number 7 is only going to come from the father's side, just 1. And the chromosomal disorder that results from that, monosomy 7, refers to that's where the chromosomal disorder has occurred. And that's Turner syndrome. And there's specific things that happen with typically females that end up with Turner syndrome. Let's look at the flip side, trisomy. So tri refers to 3. So it lets you know that polysomy, poly means many, and this refers to the fact that there are going to be 3 chromosomes instead of the normal 2. So let's look at a blow up of this one. Okay, let's focus on the circle errors again. So again, the normal, in this example, the normal egg should have one chromosome and the normal sperm should have one chromosome. So when they come together, you get back to 2. However, if you have a trisomy, you have a failure of separation. So the egg, instead of having the normal 1, has 2. So let's say that this egg with 2 chromosomes matches up with a normal sperm, which has the 1 chromosome. Now, when you have a fertilized zygote, you have 3 chromosomes instead of just 2. So again, going back to our human example, 23 chromosomes from the sperm, but let's say that the egg has 24 chromosomes. It has an extra chromosome number 21. So when the zygote is formed, chromosome number 21 has two contributions from the egg and 1 contribution from the sperm, which results in 3 chromosomes on chromosome number 21. Chromosomal disorder trisomy 21 is Down syndrome, and most of you are very familiar, maybe somebody in your family, certainly have met somebody, I'm sure, in your life that has Down syndrome. And this is a result from 3 chromosomes for chromosome 21. So that is chromosomal disorders. So again, I just want to go back to this slide really quickly. And the things that you're going to need to understand is what the normal count should look like in the 4 eggs or the first 4 sperm. And then what would happen if they came together to form a zygote? Would you have a monosomy or would you have a trisomy? And that is chromosomal disorders.